Dear students, Dr. Muhammad Irfan, and today we are gonna discuss experiment number 7. The topic of the experiment is to observe the process of diffusion and the effect of temperature on the rate of diffusion by using K. By the end of the practice should be able to 1. Demonstrate process of diffusion and effect of temperature experimentally using KMnO4 crystals. 2. Define terms like active and passive transport, simple and facilitated diffusion and osmosis. 3. List factors increasing and decreasing the rate of diffusion, and 4. Plot a graph to show effect of temperature on rate of diffusion. The requirements are KMnO4 crystals, Petri dishes, pencil, ruler, plain white paper, normal, cold and hot, tap, water, measuring cylinder graph. First of all, we will talk about different modes of transport across cell membrane. There are basically two types of processes are involved in transport across the cell membrane. 1. Passive transport, which is the movement of molecule from higher concentration to lower concentration without utilizing the energy of ATP, and 2. Active transport, which is the movement of molecule from lower concentration to higher concentration by carrier protein, utilizing. In this slide, you can see different types of passive transport. Remember, all will follow the same rule of moving from higher to lower concentration gradient without using the ATP as their energy source. There are two types of passive transport, diffusion and osmosis. Diffusion, which is a type of passive transport, is again divided into simple diffusion that is carried out without taking any help from carrier transporter protein, and facilitated transport, which is carried out by using the help of a carrier transport protein. Remember, both of these are types of passive transport and diffusion, but the difference between them is the carrier protein that limits the transport of molecules even if there is a big concentration gradient, as it can transport a limited number of molecules one time.
So basically, occurs because of a random molecular movement of a substance from an area of their higher concentration to lower concentration, as shown in the picture.
Simple diffusion, as I told you, it is the movement of molecules in the direction of their concentration gradient by themselves using their kinetic energy. Examples are diffusion of oxygen from alveoli to the circulating blood and carbon dioxide from blood to alveoli. Facilitated is also the movement of molecules in the direction of their concentration gradient, but with the help of a carrier protein molecule. The best example is the movement of glucose. Rate of diffusion is defined as the number of molecules moving from an area of higher to lower concentration per unit time. Unit time may be a second, a minute or an hour. Some factors will increase or decrease the rate of. This slide shows increasing the rate of diffusion. These are 1. Concentration difference, that is, greater the difference of number of molecules between higher and lower concentration sites, the greater or faster will be the rate of diffusion. 2. Temperature. Means increase in temperature, increases the kinetic energy of moving molecule, increasing the rate of diffusion, and 3. Surface area, that is, larger the membrane's surface area for diffusion. This slide shows decreasing the rate of diffusion. These are 1. Thickness of membrane. Means, thicker the membrane the slower will be the movement of molecules across it. 2. Molecular weight. Means, greater the molecular weight of a substance used for diffusion, the slower will be the rate of diffusion, and 5. Distance. Means, greater the distance required. Here the procedure, the experiment is shown. The procedure starts with the placement of one crystal of KMnO4 in the middle of the Petri dish containing water, that may be normal tap water, warm, or cold water. Immediately after placing, KMnO4 starts diffusing from the middle of the Petri dish towards periphery. After every 1 minute, measure the diameter of the circle for the next 3 minutes and write the readings in the table provided. Repeat the same procedure with.
In this slide, some observations are given for the diameters after every 1 minute. Next slide shows the same for cold water and warm water respectively. You need to plot a graph, taking time upon x-axis and measurement upon y-axis.
Dear students, the experiment. I hope you have followed it properly. Thanks a lot and bye.